Flowers are the site of sexual reproduction in angiosperms. The androecium consists of stamens, while the gynoecium consists of pistils. This presentation will explore the fascinating process of sexual reproduction in flowering plants, from pollination to seed development. The anther, a crucial part of the stamen, typically has 2 lobes, each containing 2 pollen sacs. It consists of 4 wall layers: epidermis, endothecium, middle layers, and tapetum, which surround the microsporangium. The sporogenous tissue in the center undergoes meiosis to form microspore tetrads. Pollen grains develop through a series of stages. Initially, microspore mother cells undergo meiosis to form tetrads of haploid microspores. These microspores then develop into pollen grains with a 2-layered wall, outer exine and inner intine. Mature pollen grains contain 2 to 3 cells, including vegetative and generative cells. The pistil is composed of 3 main parts: the stigma, style, and ovary. The stigma receives pollen grains during pollination. The style connects the stigma to the ovary and provides a path for pollen tube growth. The ovary contains ovules, which develop into seeds after fertilization. The ovule is a complex structure consisting of several parts. The funicle attaches the ovule to the placenta. The integuments are protective layers surrounding the nucellus, which contains the embryo sac. The micropyle is an opening in the integuments for pollen tube entry. The development of the female gametophyte begins with megasporogenesis, where a megaspore mother cell undergoes meiosis to form 4 megaspores. Typically, 3 megaspores degenerate, and 1 functional megaspore develops into the embryo sac. The mature embryo sac is a 7-celled, 8-nucleate structure. Pollination is the transfer of pollen grains from the anther to the stigma. It can occur through abiotic agents like wind and water or biotic agents such as animals, including insects, birds, and bats. There are different types of pollination. Autogamy refers to self-pollination within the same flower. Gatonogamy is pollination between different flowers of the same plant. Xenogamy involves cross-pollination between flowers of different plants. Pollen-pistil interaction involves several steps. Pollen grains land on the stigma, which recognizes compatible pollen. The pollen then germinates and forms a pollen tube, which grows through the style to reach the ovary. Double fertilization is a unique process in flowering plants. One male gamete fuses with the egg cell to form a zygote, while the second male gamete fuses with 2 polar nuclei to form the primary endosperm nucleus. After fertilization, several events occur. The triploid tissue forms to nourish the developing embryo. The zygote develops into an embryo through various stages. The ovule matures into a seed with an embryo, endosperm, and seed coat. Finally, the ovary develops into fruit, enclosing the seeds. Endosperm development involves 3 stages. In the free nuclear stage, the primary endosperm nucleus undergoes repeated divisions without cell wall formation. During cellularization, cell walls form around free nuclei, creating cellular endosperm. Nutrient storage accumulates reserve food materials for embryo nutrition. Embryo development progresses through several stages. The proembryo stage follows the early division of the zygote. The globular stage forms a spherical mass of cells. The heart stage gives the embryo a heart-like shape. Finally, the mature embryo is fully developed with cotyledons and an embryonic axis. The structure of a mature embryo varies between dicots and monocots. Dicots have 2 cotyledons, an epicotyl, hypocotyl, and radicle. Monocots have a single cotyledon called the scutellum, along with a coleoptile, coleorhiza, and radicle. Seeds have a protective outer layer called the seed coat, derived from integuments. Inside, the endosperm provides nutrition, and the embryo contains cotyledons and an embryonic axis. The micropyle is a small pore for water entry during germination. Seeds can be classified into albuminous and non-albuminous types. Albuminous seeds, like wheat and maize, contain residual endosperm. Non-albuminous seeds, such as peas and groundnuts, lack endosperm, with food stored in cotyledons. Fruits can be categorized into true fruits, false fruits, and parthenocarpic fruits. True fruits develop only from the ovary, like mango. False fruits develop from the ovary and other floral parts, like apples. Parthenocarpic fruits develop without fertilization, resulting in seedless fruits like bananas. Apomixis is the formation of seeds without fertilization. It can occur through mechanisms like diploid egg cell development or nucellar embryony. Apomixis is important for maintaining hybrid vigor in successive generations. Polyembryony is the phenomenon of multiple embryos developing in a single seed. This can occur through various mechanisms, including the division of the zygote or development from synergids or antipodal cells. Typically, only one embryo successfully develops into a seedling. Sexual reproduction in plants has several significant benefits. It creates genetic variation, enhancing adaptability. It allows for natural selection and species evolution. It enables breeding programs for crop improvement and contributes to the rich diversity of plant species on Earth.